Okay. So these are some examples of transmembrane proteins that, that we will be coming across quite often in the lectures in the future. So there are many, many types of transmembrane proteins that have both functional and structural differences. And each transmembrane protein is like, if we can consider it like a machine. So they have their own functions, and different machines can carry out different functions. So as the first example, you have the example of a protein called rhodopsin. Rhodopsin is a protein that is found in our retina. It consists of a protein component that is membrane bound, and it also has a small group which is made of vitamin A. So this is a component that gives us the ability to receive light and to recognize light, to be activated in the presence of light. So when it detects light, it's a 7 transmembrane alpha helical structure. So this is a G protein coupled receptor, an example of a G protein coupled receptor. It's 7 transmembrane alpha helices. They shift position relative to each other, and this creates an intracellular signaling cascade. So let's have a look. So it receives the light, and then when it receives the light, it undergoes a conformational change, which can create a signaling transduction. This signal will then transfer to the brain, and it will allow us to see. Another example, very important example, is of the EGFR, which can bind to epidermal growth factor, which is the ligand. And when it binds to the EGF, you can see that the 2 domains will come close to each other. They will dimerize, and it will lead to the activation of an intracellular cascade. So here, this is an example of how receptors can move along a membrane. And remember, we were talking about it yesterday on how movement along the membrane is very easy, but movement across the membrane is very difficult. So this fluidity of the membrane allows the 2 halves of this protein to come together only when the ligand is present. In this case, the ligand is the EGF. Only when the ligand is present, these 2 proteins come together. And when they come together is the only time when the kinase domains, EGFR is a kinase, when the kinase domain gets activated, causing phosphorylation of the EGFR itself. So it uses ATP to phosphorylate EGFR, which will then activate a signaling cascade that will lead to further downstream signaling from the EGFR. There's one other interesting point that has been made in the previous protein, that example of protein, which is rhodopsin, and this one as well, in that the part of the membrane that is the part of the protein that is located in the membrane is said to have an alpha helix. So here you can see activation of the intracellular kinase domain results in the start of an intracellular signaling cascade that eventually triggers cell to grow. EGFR possesses a single transmembrane alpha helix. So the alpha helix is exactly the same alpha helix that we have come across previously when we were talking about the secondary structures of proteins, the alpha helix and the beta sheet. So the alpha helix, in this case, is the helical structure where the presence of the amino acid will be organized in such a way that those faces that are facing the hydrophobic domain will have more hydrophobic amino acids. I will explain this more later on. Cadherin, this was a beautiful, beautiful picture. Cadherins are cell-cell adhesion proteins. Okay, so this is one membrane, this is another membrane, and between 2 membranes, we have these cadherin proteins. One example that some of you or many of you may be familiar with are the E-cadherins. So E-cadherin or cadherins, it's a big family of proteins. In the presence of calcium, the extracellular domain, so this is outside the cell, the extracellular domain of the cadherin from one cell binds to the extracellular cadherin domain of the adjacent cell. So this allows for cell-cell interaction to happen. So between the membranes, there is interaction, which allows for signaling to take place. The internal domain, this is the extracellular domain. The intracellular domain, which you can see if you can follow my cursor, the intracellular domain is bound to the actin cytoskeleton. So this is how outside-in signaling can happen as well, whereby the cadherins binding to other cadherins on an adjacent cell is going to lead to the activation of intracellular signaling through cytoskeletal proteins such as catenins as well as actin, and the actin filaments will then take this signal to the nucleus and it will give various different messages to the cell. For example, it can give the message that there is a neighboring cell, so you are okay, you can keep the survival signals, you can keep growing. So here we have it. It's really, really nice. So we have an intracellular domain which is bound to the actin. So cadherin proteins provide a structural connection point between cytoskeletons of adjacent cells. And they also have a transmembrane domain that is a single alpha helix. And then the last example that we have here is the ATP synthase. We've already come across the ATP synthase previously. It's a very elaborate protein, more than 20 polypeptides, and it maintains this asymmetric distribution of protons across a membrane. In the previous lecture, we also learned how the presence of this ATP synthase causes a bend in the cristae, right? So the presence of both of these subunits of the ATP synthase is always found at the edge of this cristae. And this basically makes sure that there is a bend in the membrane as well. So why do I mention about the bend? Is because when we have a protein in the membrane, that protein is not there just to carry out the function of the protein, such as binding to EGF and sending signals into the cell, but it can also determine the shape of the cell. So the presence of the protein and the presence of the phospholipids, both of them, can determine the shape of the cell as well. It's not just a question of function, it's also shape. Okay, now, are there any questions so far? If there is anything to ask, I will be happy to address them. If not, I'm going to start with the PowerPoint slide and talk a little bit more about membranes, and then we will move on to our next topic.
Tom, there is a question.
Go on.
Why hydrophobic molecules?
Okay. So when we are when we look at all these membrane-bound proteins, and with the whiteboard, I tried to explain to you how a channel like this may be structured in a very, very hydrophobic environment of the membrane. So we have the hydrophilic head groups here, but we do have a very, very strongly hydrophobic environment in the fatty acid tails. So these sort of transporters that we are going to be learning about now, they tend to have several alpha helices that sort of form a tube-like structure, a channel across the membrane, which, because of their hydrophobic and hydrophilic side chains of the amino acid that form these transporters, they can survive very nicely in the amphipathic nature of this particular of a membrane. So when we are discussing about transport across membranes can be either passive meaning no energy is required for the transport to occur, or it can be active, which means energy is required for the transport to occur. In the case of passive transport, we can either have non-mediated transport, or we can have transporter-mediated transport. Now, these two, the B and C, they look very, very similar, but there are some subtle differences between them, which I would like to draw your attention to now. You look at this particular transporter, it seems to be open all the time. But this transporter appears to have an occlusion here. It appears to be closed a little bit, right? So keep this in mind, and I will come back to it later once we've talked about how passive diffusion, passive movement can happen. For passive movement to happen, in other words, for a molecule to move across a membrane without the use of any energy, we have to have a concentration gradient, right? So that's the most important thing. We have to have more of the molecule outside the cell. In this case, if we follow the arrow of the movement, and if we consider that this part of the membrane that I'm pointing with my cursor is the extracellular portion, and the intracellular portion is across the membrane, then we have to have higher amount of the molecule outside the cell for it to move passively inside the cell. Now, only concentration is not enough in many cases. Concentration is always important, but it is not the only factor. For example, between two molecules that have very, very comparable concentration gradients, in other words, both molecules are found in higher amounts outside the cell, but you end up seeing that one of them moves into the cell more efficiently. So this could be related to the fact that those particular molecules that are moving across the membrane more efficiently may be more hydrophobic in nature, right? So we have to also consider the partition coefficient, whether they dissolve more easily in oil or more easily in water. If something dissolves more easily in oil, such as estrogen, a cholesterol-based hormone, then it will pass across the membrane very, very easily without expecting any transporter. It does not require a transporter. If it is a very small molecule, such as oxygen or carbon dioxide, and it's uncharged, it will also move very, very easily. It will just slip between the spaces that we have in the phospholipid bilayer, and it will move very easily. On the other hand, if it is something that does not dissolve well in oil, okay, if it is something that is highly hydrophilic, then its passage across the membrane, even in the presence of a high concentration gradient, will be reliant on the presence of a transporter. This is the point that I was trying to make earlier with Zeynep's question, that even if there is a high concentration gradient, in the absence of a transporter, a hydrophilic molecule is unable to pass the membrane simply because of the presence of this very, very, very severely hydrophobic interior of the membrane. In this case, we will have a transporter. And in this case, the transporter can be, for example, an ion channel, right? So if we consider molecule B to be an ion, such as sodium ion, it's always easier to imagine sodium or calcium or chloride, because these are the ions that are found in greater concentration outside the cell compared to the inside. Then these can move very easily across the membrane as long as there is a concentration gradient available to them. This movement does not require any energy. Sometimes there are ion transporters that can allow the movement of any positively charged ion. Sometimes there are very, very specific ion transporter. For example, a sodium transporter will only transport sodium, it won't transport anything else. Potassium transporter will only transport potassium and nothing else. So there are specific ion channels, and there are relatively non-specific ion channels that will allow only the similarly charged, so a positively charged ion, to pass through. We don't have any ion channels that allow positive as well as negative ions to pass through, clearly because the charge on the ion will, the movement of a charged ion across a transporter will depend on the amino acid side chains that are found at the edge where the transport is happening, right? Inside this channel where the transport is happening. So if you have a positively charged side group here, then a negatively charged ion can be passed easily. It won't be repelled. But if you have a negatively charged ion here, if you have a negatively charged side group here, a negatively charged ion cannot pass. So therefore, depending on the charge, you can have cation channels or you can have anion channels, but you cannot have both cation and anion passing through the same ion channel. Okay? When we come to transporter mediated can either be passive or it can be active. So what we mean by a passive transporter mediated is, in this case, the transporter is very, very, very specific to the molecule that it is transporting. So in order to understand it better, it would be best to start with an example, such as the glucose transporter. The glucose transporter is very specific to glucose. It is very specific to the isomer of glucose. So a D isomer or an L isomer, it will only recognize one isomer of glucose. So it's that specific. In this case, what is happening is the transporter can move glucose in either direction. It can either move it from the extracellular to the intracellular environment, or it can move from the intracellular to the extracellular environment. It can do both, provided there is a concentration gradient. So if inside the cell we have a lot of glucose, it will move, the transporter will move the glucose to the outside of the cell. But if we have a lot of glucose outside of the cell, the transporter will move the glucose inside of the cell. So this glucose transporter, which is an example of facilitated diffusion, this is basically an example of, it works very, very similarly to an enzyme. So just as an enzyme has a very strong structural affinity to its cognate substrate, it will only bind to its substrate and nothing else. Facilitated diffusion also is very, very specific for the molecule that it is transporting. So a glucose transporter will only bind to glucose and move it in a specific direction, depending on the concentration gradient. So just as a quick caveat of how this works in our cells, as soon as glucose enters our cell, in order to maintain this concentration gradient of glucose, glucose is immediately converted to glucose 6-phosphate. It is phosphorylated in the presence of an enzyme called hexokinase, which converts glucose to glucose 6-phosphate. As soon as glucose is converted to glucose 6-phosphate, it now becomes something else, right? It's not glucose anymore. And so we maintain a concentration gradient of glucose, and glucose keeps coming inside the cell. In general, most of the cells in our body they don't have enough free glucose inside the cell in order to have the movement in the opposite direction. Most of our cells need glucose for energy production, and therefore the glucose will be coming inside the cell. But there is one organ in our body which is capable of moving the glucose in the opposite direction. And I would like you, either using the chat box or opening up your microphone, to tell me which organ you think is capable of doing this. Liver, bravo. Oh, okay, very, very good. So liver, indeed, is the organ where we are capable of moving glucose in the opposite direction, because liver stores the glycogen for the rest of the body. Now, you may ask at this point that glycogen is also stored in skeletal muscles. Indeed, glycogen is stored in skeletal muscles, but that glycogen, when it's converted to glucose, is used by the muscle itself. It is not released into the blood because the muscles are contracting and they need the energy from the glucose. But when it comes to the liver is a very selfless organ. It's a very, very good organ for us, right? So it stores the glycogen, and then when our body needs this glycogen, it releases the glucose from the glycogen to the circulatory system, and then this glucose can be taken to the essential organs such as our brain. So indeed, in the liver, we can have movement of glucose in the opposite direction as well, which is the beauty of the glucose transporters that we have. Another quick point that I would like to make here, and before we move on to the active transport, is that there are several types of glucose transporters that we have, GLUT1, 2, 3, 4, etc. And you probably, some of you or all of you already know that the presence of a glucose transporter in the membrane is often reliant on the presence of insulin. So the only glucose transporter that is responsive to insulin, the only glucose transporter that will be moved to the membrane when insulin signaling is found, is GLUT4. Okay, so the insulin mediated intake of glucose usually occurs when storage of glucose is necessary. Okay, so you've just eaten a meal, you have your blood sugar is high, in this case insulin secretion starts, and insulin will go and bind to insulin receptors on certain cells which will take in the glucose and store the glucose. For those cells that are using the glucose, you don't need GLUT4. You don't need insulin. Those cells always have a glucose transporter such as GLUT1 or GLUT2, which will allow glucose to move in as and when they need the energy. But as soon as you've eaten a carbohydrate-rich meal and you have a lot of glucose in your blood, and this glucose now needs to be stored either as glycogen or as fat, then we will need to have a glucose transporter that is responsive to insulin. In this case, the GLUT4 is expressed only in skeletal muscle, which can take in the glucose and store it as glycogen, or it is expressed in adipose tissue, in our fat tissue, which can take in the extra glucose and it will store it as fatty acids, as lipid droplets, in fact, as triglyceride, not as fatty acid, but as triglyceride. The liver has GLUT2. Liver does not have GLUT4. So liver glucose transporter is not responsive to insulin because remember, liver has to have the ability to move glucose in either direction. Okay, so it is not reliant on insulin in order to, it should not have such a regulated glucose transporter availability. This is a very important point that I would like to draw your attention to. The moment you have regulation, the moment you have a hormone that is regulating the presence or absence of a transporter, you can have two situations. You can have it present or you can have it absent. For the liver, absence of the glucose transporter is not an option because the liver always has to be ready to remove glucose from itself into the bloodstream in order to provide glucose for the brain, right? So therefore, if it had a regulated glucose transporter, it would need to wait for that signal to come, for the glucose transporter to be expressed, to be moved to the membrane. By then, the organism is dead, right? So therefore, there is no regulation of the glucose transporter in the liver. It's always present. And it will move the glucose in whichever direction, outside to in or inside to out, depending simply on the concentration gradient. Okay, so now we will come to the last part of this, of the membranes, which is a discussion on how active transport happens. Active transport happens only when there is movement against a concentration gradient. So in this case, we need the energy, and you've already come across an example of active transport when we were looking at the ATP synthase. Remember, we had the pumping of the protons across the inner mitochondrial membrane. That pumping, remember I said, please be careful about the use of the word pump, because pump is used only when the transport is against a concentration gradient and ATP is utilized. Otherwise, it's a channel. So a pump is something that uses energy in order to move an ion or any molecule against a concentration gradient. So here we have low concentration of an ion outside of the membrane, high concentration of the ion inside the membrane. But if we need to move this ion, then we will need to utilize energy. So an example of DE would be potassium ions. Potassium ions are found much more inside the cell. Potassium ions are found a lot less outside the cells. So to move potassium across the membrane, we will need to utilize energy. A similar example will be a calcium ATPase. The calcium ATPase is found in many, many cell types, but it's especially important in muscle cells because the calcium it helps in the contraction of the muscles. It helps the actin and myosin to interact with each other. And when the actin and myosin does not need to interact with each other, when the muscle is relaxing, then the calcium needs to be moved into the sarcoplasmic reticulum, which is the endoplasmic reticulum of the muscle cell. And that movement is also against a concentration gradient. So we need energy there in order to move the ions. So now we are going to, okay, so we have some of the examples here. I'm going to come back to this later, but I would like to talk about the sodium-potassium ATPase. So why is energy needed for a sodium-potassium ATPase? And what is a sodium-potassium ATPase? Let's come to that first. Sodium-potassium ATPase is an active transporter that moves 3 sodium ions from the inside of the cell to the outside and 2 potassium ions from the outside of the cell to the inside. Okay, so 3 sodiums leaving the cell, 2 potassiums entering the cell. In both cases, we are moving against a concentration gradient. Remember, we have more sodium outside the cell, more potassium inside the cell. So either movement, sodium out or potassium in, is against a concentration gradient, which is why we require energy. But what exactly is this energy doing? To appreciate what this energy is doing, you need to appreciate what this protein is capable of doing. This protein, the sodium-potassium ATPase, has to have high affinity for sodium in an environment where there is less sodium. Okay, so inside the cell, remember, we have relatively fewer sodium ions. So it has to have very high affinity for sodium here. As soon as it binds to the sodium, it will need to release the sodium to the extracellular space. So while it had high affinity for the sodium ion on the intracellular phase, its affinity for sodium ion on the extracellular phase has to be very, very low. Because it has low affinity here, it will release the sodium, right? And now when it has low affinity for sodium in the extracellular phase, it also has to have high affinity for potassium here. So it can bind to the potassium, and this potassium will then move to the intracellular phase, and then this protein has to lose its affinity for potassium in order to release it. So there is a lot of changes that this particular protein has to go through in order to bind to the sodium, to release it, to bind to the potassium, and to release it. And all of these changes are possible by the breakdown of ATP into ADP and into PI. So let's have a quick look at how this is working. There are two conformations of this protein, the E1 conformation and the E2 conformation. And you can see from this cartoon that the shape of the protein is very different in the E2 conformation compared to the E1 conformation. We also learned in a previous lecture that the phosphorylation event, which as we know is a post-translational event, can change the conformation of a protein tremendously. And in this case, we are using the phosphorylation of an aspartic acid residue. So this is also something very unique, because normally we have serine, threonine, and tyrosine that can be phosphorylated. In this case, we have an aspartic acid residue that gets phosphorylated here. When the protein is in the E1 conformation, it has high affinity for sodium in the intracellular space. The extracellular space now is blocked. So there is like an occlusion here so that the sodium cannot escape. In any case, the sodium cannot escape because this is against a concentration gradient. The structure of this channel is such that it can allow at any time 3 sodium ions to bind. As soon as the 3 sodium ions are binding, this is a kinase, the ATP sodium potassium ATPase, will cleave the ATP and a residue gets phosphorylated. As soon as this residue gets phosphorylated, this protein now moves to an E2 conformation. So the phosphorylation event is necessary to change the shape of this protein. And in this new shape, the E2 conformation, the protein has low affinity for sodium, and it releases the sodium, the 3 sodium ions that could be accommodated within the structure. These 3 sodium ions are now released. And in this same E2 conformation, it is also capable of binding to potassium ions. Now, again, the structure of the protein is such that it can only allow 2 potassium ions to bind at any time in an energetically favorable conformation. And in order for the potassium to move and to be released into the extracellular space, into the intracellular space, the phosphate group has to be removed. So the phosphate group that was bound to the aspartic acid is now released. And as soon as it is released, this protein will go back very quickly to its E1 conformation, and it will release the potassium ions into the intracellular space. So the ATP is here to move this protein from its E1 to its E2 conformation so that the release of the sodium can happen and the binding of the potassium can happen. And from the E2, it moves back to the E1 conformation in order for the potassium to be released into the intracellular space and then for the sodium to go and bind. An important point to make here is, of course, is the fact that we have 3 sodium ions leaving and 2 potassium ions entering. So this has enormous implications in maintaining the polarity across membranes. So as you know, in neuronal cells, we have to maintain a polarity across the membrane. The inside of the cell is usually relatively more negative. It's about -70 millivolts compared to the outside. So the inside of the cell is more negative compared to the outside. And it will be easy for us to imagine why the inside of the cell is more negative, because we have 3 positive ions leaving and 2 positive ions entering. So this makes the inside of the cell relatively more negative compared to the outside. So what we have is the sodium potassium ATPase is also electrogenic. In other words, it also maintains an electrical gradient across the membrane. Okay, so I'm now going to go back just to give you a very, very quick idea of movement of water, and then I'm going to stop and we can discuss a little bit, if you have any questions. So far we have looked at the movement of ions. We have looked at passive movement of ions, we've looked at active movement of ions, and we've also looked at facilitated diffusion, which you can also look at over here. There is a nice picture, but it doesn't tell us anything more than what I have already discussed, so you can just look at it at your own time. But for the movement of water, we need to understand the concept of osmosis. Osmosis is the process of movement of water from a region of its higher concentration to a region of its lower concentration. This seems very, very simple to you, right? So yes, of course, we know what osmosis is. We've been learning about osmosis since we were in our middle school. But you also need to appreciate how very, very important the movement of water actually is. The process of osmosis is what guides the movement of water in our large intestine. If we did not have osmosis in the large intestine, we would have severe diarrhea, which is what happens when you release a lot of liquid in your feces, and that's what diarrhea is, right? There is no reabsorption of water. It would lead to severe dehydration in the human body. Osmosis is also the process by which we are reabsorbing water in our kidneys. So osmosis, therefore, is extremely important for many, many, many other functions of an organism. So just to give you a very quick idea of how osmosis works, this is a red blood cell. And when the red blood cell is put in a hypotonic solution, in other words, the ion concentrations outside the cell in the solution is low compared to the inside. So in this case, the water is understanding that there is less water inside the red blood cell because outside it's more dilute. Let's say it's a dilute solution that we've put it in. So there is some movement of water out, but there is more movement of water in, okay? Because the water wants to go and it wants to normalize the ion concentrations across this membrane. So in this case, the cell will swell very much. And in fact, for the red blood cell, this is routinely used in order to burst the cells, to release the contents of the cell, and just to have the erythrocyte membranes, which is called the erythrocyte ghost. Okay, so this is used routinely in the laboratories to generate red blood cell membranes, which are then used for research. If we have the cell placed in a hypertonic solution, in other words, there are a lot of ions outside the cell, now the water will tend to leave the red blood cell. And in this case, the red blood cell will shrink considerably. What we have in general in our body is an isotonic solution. So isotonic saline, you've probably heard this word before, whereby you maintain the ion concentration that we have in our plasma. In this case, the amount of water getting in is equal to the amount of water getting out. So we have a sort of equilibrium here, and the shape of the red blood cell remains as it should be. It maintains its normal shape. So we have no net loss or gain. Osmosis is extremely important in plant cells because plant cells do not have a skeletal system like we do, right? They don't have any bones. So the only way in which they can maintain the turgor pressure, which is basically the cell is filled up with water so that it can maintain their shape, is by the process of osmosis. So they maintain a high concentration of various different organic molecules inside the cell, which will allow water to constantly enter into the cell, which will then keep the turgor pressure high, and it will allow the plant to remain upright. So you've probably all noticed that when you forget to water your plants, they become a bit droopy, right? So the leaves are becoming droopy, the plant itself may bend, and then when you add the water, it becomes upright again. So in the absence of water, the osmosis will not happen, and the turgor pressure is therefore lost, and the plant wilts. That's called wilting of the plant. But then when you add the water again, the plant becomes upright again. Okay, so now we can have again a quick discussion before we go on the break.